Let's now look at how the mRNA vaccine works in more detail. I'm going to use my own diagram to illustrate this. The mRNA in the vaccine is short-lived and is synthesized in the lab. So basically, what they did was to get the mRNA from the S protein, which was the main problem in COVID-19. The mRNA is encapsulated in a lipid nanoparticle to reduce its degradation by enzyme that will render it ineffective, and also to increase its uptake by cells of the body, so that once injected, it can hide from the body's immune system. Now, this mRNA can be taken up by basically any cell in the body, but the immune system cell, for instance, dendritic cells, take it up more avidly compared to non-immune cells. Now, when it reaches the cytoplasm of the immune cells, the ribosomes will get to work and start translating the message carried by the mRNA into the spike protein of the coronavirus. These proteins are now the antigens. The cells display the spike protein piece on their surfaces. The immune system recognizes the spike protein as foreign and begins an attack by activating other immune system cells such as T cells, cytokines, and B cells, which will lead to the production of antibodies specifically targeted to the spike protein of the COVID-19 virus, just like what happens if one is infected with COVID-19 disease. This basically trains the body to respond when the virus is contracted in the future, leading to immunity. The benefit of using mRNA to have host cell to produce the spike protein is that mRNA is far easier for vaccine creators to produce than antigen proteins or attenuated or killed viruses. Now I need to clear some circulating misconceptions surrounding this vaccine. It is important to know that the proteins cannot assemble to form a virus. Also, the mRNA in the vaccine only works in the cytoplasm and never enters the nucleus of the cell to interact or modify with our DNA in any shape or form. Once the mRNA has passed on the message about this protein production and the proteins are produced, the cell breaks down the mRNA and gets rid of it. So the mRNA are normally degraded within a few days. We need to dive into the clinical trials data. I'm going to make it as simple as possible as it's very complex. This information is available on the MHRA website, by the way, and it's accessible and available to anyone. The vaccine has been deemed safe and effective by the MHRA. It is the first global medicine regulator in history to approve an mRNA vaccine. According to the CEO, June Rain, who said no corners have been cut in approving this vaccine and that the benefit outweighs any risk. Okay, so in phase 1 safety studies of the COVID-19, this involved 90 healthy volunteers from ages 18 to 85 years old. But we're going to dwell more on phase 2 and 3 trials. Phase 2 and 3 studies usually include volunteers that reflect the population for whom the vaccines are intended. This means enrolling people from diverse geographical areas, racial and ethnic backgrounds, genders, ages, as well as those with underlying health conditions that put them at higher risk of COVID-19 disease. Including these people in clinical trials is the only way to make sure that a vaccine will be safe and effective for everyone who needs it. In phase 2, there were 360 volunteers from ages 18 to 85 years old. In phase 3 studies, approximately 44,000 candidates were involved. This includes the 360 from phase 2. Phase 2 and 3 were done side by side, and this study later included ages from 12 years old. Now, concerning the dose, 2 doses were given, while there was some evidence to indicate high levels of short-term protection from a single dose of the vaccine. A 2-dose vaccine schedule is currently advised, as this is likely to offer a longer-lasting protection. Total participants were 43,651, although this number decreased over time, but not majorly though, due to some of them being excluded and some did not even show up at all for the study. About 50-50 of the candidates received 30 micrograms of the Pfizer-BioNTech vaccine, and the placebo group got saline. The candidates for the vaccine were roughly 50-50 male to female ratio who received the vaccine and placebo, which is great. When we look at the ratio uptake of this vaccine, about 83% of them were white compared to others. Black and African American were only 8.8%, Asians were 4.4%, and others were even much lower, as you can see. The good thing though was that at least some races were covered, but clearly the population for these clinical trials wasn't diverse enough. And of course, we can say it's not a true representation of the global population that will receive this vaccine. Certainly, the minorities were mostly affected due to socio-economic and environmental factors, but unfortunately, the majority of the minority groups were unwilling to partake in these trials, hence the result we see. In addition, this trial was carried out in Argentina, Brazil, Germany, South Africa, and the USA. In the study, those who received vaccination with the mRNA had a reduction in the rate of COVID-19 illness compared to those who received the placebo. 8 cases of COVID-19 illness in the vaccinated group compared to 162 cases in the placebo group. These infected candidates had no evidence of COVID-19 disease before the vaccination program and after 7 days of receiving the second dose. These people were not intentionally exposed to this virus, by the way, but contracted it by going through the usual daily activities like everyone else. This result translates to 95% efficacy of the vaccine in those ages 16 years and over. Data from the trials indicate high vaccine efficacy, high tolerability with no serious safety concerns observed. Efficacy was consistent across age, gender, ethnicity, and in the presence of all the comorbidities. That means all the illnesses, including asthma, obesity, diabetes, hypertension, and lung diseases. Now, there is a need to address people of color like me because there is a lack of trust and skepticism in taking the COVID-19 vaccine, mainly due to safety and effectiveness. When we look back at history, for instance, let's look at the unjust medical treatment among the minorities in the Tuskegee syphilis study, which happened in 1932, where some African American men who had syphilis were enrolled in a study. Most of these men were poor and illiterate. They didn't tell them what they were using them for. They basically tricked them into the study. The investigators knew they had syphilis, but unfortunately, they didn't tell them. And by 1945, penicillin was already the treatment of choice for syphilis, but these men were in absolute denial and never received any treatment, just for the investigators to see the effect of the disease progression on the human body. Some of these men end up dying as a result of the complications from untreated syphilis. They were basically used as guinea pigs, which is very, very sad. But today, I can assure you that so many of this would have been unethical and would never have happened in the first place. The main difference now is transparency. Nobody will give you what you know nothing about, and the data are published online for all to see and read if you're interested. Also, consent is very crucial now than in the past, which is needed before any medical treatment is given. The individual will be informed about the process, benefits, and the risks of the vaccination as well as its side effects, and your questions will be answered. The key thing we need to take home is that this mRNA vaccine had been tested on tens of thousands of people before it was rolled out for use in the wider population, which is now beginning to at least, millions of people worldwide, and so far it has been well tolerated.